MIT engineers have invented a new 3D fabrication method that can generate a novel type of drug-carrying particle that could allow multiple doses of a drug or vaccine to be delivered over an extended time period with just one injection. The new microparticles resemble tiny coffee cups that can be filled with a drug or vaccine and then sealed with a lid. The particles are made of a biocompatible FDA-approved polymer that can be designed to degrade at specific times, spilling out the contents of the cup. The researchers are very excited about this work because for the first time they can create a library of tiny encased vaccine particles, each programmed to release at a precise predictable time so that people could potentially receive a single injection that in effect would have multiple boosters already built into it. This could have a significant impact on patients everywhere, especially in the developing world where patient compliance is particularly poor. The researchers began working on the new drug delivery particles as part of a project funded by the Bill and Melinda Gates Foundation, which was seeking a way to deliver multiple doses of a vaccine over a specified period of time with just one injection. That could allow babies in developing nations who might not see a doctor very often to get one injection after birth that would deliver all of the vaccines they would need during the first 1 or 2 years of life. Using photolithography, they created silicon molds for the cups and the lids. Large arrays of about 2000 molds are fit onto a glass slide, and these molds are used to shape the PLGA cups and lids. PLGA is a biocompatible polymer that has already been approved for use in medical devices such as implants, sutures, and prosthetic devices. PLGA can also be designed to degrade at different rates, allowing for the fabrication of multiple particles that release their contents at different times. Once the array of polymer cups has been formed, the researchers employed a custom-built automated dispensing system to fill each cup with a drug or vaccine. After the cups are filled, the lids are aligned and lowered onto each cup, and the system is heated slightly until the cup and lid fuse together, sealing the drug inside. The researchers developed a new method called seal, that is stamped assembly of polymer layers, to align and seal the layers. It can make structures which current 3D printing methods cannot. In mice, the researchers showed that particles released in sharp bursts without prior leakage at 9, 20, and 41 days after injection. They then tested particles filled with ovalbumin, a protein found in egg whites that is commonly used to experimentally stimulate an immune response. Using a combination of particles that released ovalbumin at 9 and 41 days after injection, they found that a single injection of these particles was able to induce a strong immune response that was comparable to that provoked by 2 conventional injections with double the dose. The researchers have also designed particles that can degrade and release hundreds of days after injection. One challenge to developing long-term vaccines based on such particles is making sure that the encapsulated drug or vaccine remains stable at body temperature for a long period before being released. They are now testing these delivery particles with a variety of drugs, including existing vaccines such as inactivated polio vaccine and new vaccines still in development. They are also working on strategies to stabilize the vaccines.